Hello. The students who have joined, kindly can you just write in the chat box if my voice is clear and the presentation is visible to you? Please write in the chat box if my voice is clear, if you can hear my voice clearly, and if the presentation is visible to you. Okay, thank you so much. We will wait for some minutes and then we will start today's session. Okay, so let me introduce myself first. My name is Moshumidit and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am the TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today is the week 8 live session of this course. We will discuss some 10 to 15 questions today for this week 8 course content. The questions will be related to the week 8 course content, and this kind of question will help you to do your assignments and to learn and understand the course better. We will wait for some minutes and then we will start today's session. Okay. So in meantime, if you have any doubt, any queries, please write in the chat box. At the end of the session, I will answer those queries. Okay, so it's 7 p.m. So let us start the today's session. If somebody joins later, we will just allow him or her to join today's class. So we do the presentation. Okay. So today is the week 8 live session for this course of Organic Chemistry in Biology and Drug Development. The week's content in this week was the coenzymes and other vitamins. Today we will discuss the related topics, and the question related to these topics and this MCQ kind of question will help you to do your assignment for this week. Okay. So now let us just start today's session. Okay. The first question, the problem number one, it says that identify the incorrect statement among the followings. Number A, acetyl CoA is the thioester of coenzyme A and acetic acid. Option B, pyruvate is the starting point of the synthesis of acetyl CoA. Option C, pyruvate is formed by the metabolism of glycogen. And option D, TPP, that is the thiamine pyrophosphate, participates in the acetyl CoA formation. Okay, somebody wants to join. So, again, I am going to repeat the first question. The first problem is identify the incorrect statement among the following. Option A, acetyl CoA is the thioester of coenzyme A and acetic acid. Option B, pyruvate is the starting point of the synthesis of acetyl CoA. Option C, pyruvate is formed by the metabolism of glycogen. And option D, TPP or thiamine pyrophosphate participates in the acetyl CoA formation. So, what will be the incorrect statement among these following? So, let me just show you the answer first and then I will discuss this problem. Again, I am saying that if you have any doubt, any query regarding any question at any point of today's session, please do write in the chat box. At the end of the session, I will try to answer that query. Okay. So, the answer of this question will be number C. Pyruvate is formed by the metabolism of glycogen. This is the incorrect statement among these four. Now let us discuss each and every point and understand why the number C is the incorrect option. Okay, so actually acetyl CoA, the first option, acetyl CoA is the thioester of coenzyme A and acetic acid. In this figure, you can see the structure of acetyl CoA. And this part, this is the thioester part. Okay, this is called the thioester. We all know that as ester is formed by the reaction of alcohol and an acid. Okay, an alcohol and an acid. The reaction between these two give us the ester. And when this alcohol is replaced by the thiol group, this is called the thioester. Means the S double bond O, this functionality is called the thioester. So in acetyl CoA, this part is the thioester, and this thiol part, if there is it was the hydrogen here, this thiol, it will be called coenzyme A. This whole part, it is the coenzyme A part, and this moiety in the blue you can see it is derived from the acetic acid. So the first option is correct regarding the acetyl CoA. Now the second option, it says that pyruvate is the starting point of the synthesis of acetyl CoA. It is again the correct answer. Okay, because from pyruvate the acetyl CoA synthesis is happened. And in the third option, it says that the pyruvate is formed by the metabolism of glycogen. It is the incorrect statement. It will be the glucose, the metabolism of glucose, which gives us the pyruvate, and from this pyruvate the acetyl CoA formation, the acetyl CoA synthesis happens. Okay, and the option D, it is also correct, which says that the thiamine pyrophosphate participates in the acetyl CoA formation. So just remember the structure of acetyl CoA, which is consisting of coenzyme A and acetic acid, and also the starting point of this synthesis of acetyl CoA. This acetyl CoA molecule, it is very important molecule in our biology, and I think I am clear about this question. Quickly, I will move toward the next problem. If you have any doubt, please write in the chat box. Okay. So moving toward our next problem, it says that the correct state of three coenzymes involved in the biosynthesis of acetyl CoA are TPP, FAD, NADH. It is the option A. Option B is TPP, which stands for thiamine pyrophosphate, lipoic acid, and FAD. Option C is FAD, NAD plus, and lipoic acid. And option D is lipoic acid, NADH, and TPP. So we have to identify the correct state and the correct order of these three coenzymes which are involved in the biosynthesis of acetyl CoA. I will first show you the answer, then we will discuss this biosynthesis of acetyl CoA and the role of each and every three coenzyme. So the correct state of three coenzymes involved in the biosynthesis will be option B, TPP, lipoic acid, and FAD. Now let us discuss. Okay, so the conversion of pyruvate to acetyl CoA, it is the pathway from which the acetyl CoA biosynthesis happens. Okay, the pyruvate is the starting point, and from that we get the acetyl CoA. And as you can see in this figure, that the pyruvate, it goes to acetyl CoA, and the delta G negative of this delta G value, the free energy value is minus 33.4 kilojoule mole inverse, means it is a spontaneous reaction. Okay, so the various elements of this reaction are hence like like coenzyme A, which is needed because the acetyl CoA is the thioester of this coenzyme A, and there is a NAD plus involved here. This TPP, lipoic or lipoic acid, and FAD, these are the three coenzymes which are involved in this reaction. And pyruvate dehydrogenase, this complex, this actually catalyzes this reaction. Okay, this is the pyruvate, and after the decarboxylation, that is the elimination of carbon dioxide from this pyruvate, this acetate moiety, it is transferred to the coenzyme A, and the acetyl CoA formation happens. Okay, so the net reaction, it is the oxidative decarboxylation of pyruvate. Oxidative decarboxylation because the carbon dioxide is eliminating, and this is an oxidative reaction. Okay, the first carbons of glucose to be fully oxidized, that is why it is called the oxidative decarboxylation. And this reaction is catalyzed by PDH. PDH stands for pyruvate dehydrogenase, and it requires 5 coenzymes. Actually, not five coenzymes. It requires 3 coenzymes, and the coenzymes are TPP, lipoate, FAD, lipoate or lipoic acid, and FAD. These are prosthetic groups, and NAD plus and coenzyme A. These are the co-substrate. Okay. So, what is the role of this all coenzymes? We will discuss in the next questions. And I think this is the clear from this slide, that what is the name of the 3 coenzymes. Just remember the names of these 3 coenzymes, and what is the role of these 3 coenzymes. You need to remember that too, and that we will discuss in the next slides. Okay. So the correct option will be option B. TPP, lipoic acid, and FAD. These 3 coenzymes are involved in the biosynthesis of acetyl CoA. Okay. So now we will move toward our problem number 3. Again, I am saying if you have any doubt, please write in the chat box. At the end of the session, I will answer those doubts. Okay. Okay. Now the problem 3, it says that the roles of TPP and lipoic acid in formation of acetyl CoA are option A, decarboxylation and transesterification, option B, transesterification and oxidation, option C, dehydrogenation and alkylation, and option D, decarboxylation and transamination. So again, in the previous slide, I have said you that the TPP, lipoic acid, and the FAD, these 3 coenzymes are involved in the acetyl CoA biosynthesis. And I also told you to remember the roles of these particular 3 coenzymes in this process. And in this question, we will discuss the roles of this in detail. Okay. So first, I will show you the answer. Then we will discuss the biosynthesis of acetyl CoA from the pyruvate. So the correct answer of this question, or that the number 8, decarboxylation and transesterification. That is the role of TPP, the thiamine pyrophosphate, in the biosynthesis of acetyl CoA is the decarboxylation, and the role of lipoic acid in the formation of acetyl CoA is the transesterification. Now let's quickly move toward the figure. Yes. So this is a schematic diagram of the formation of acetyl CoA from the pyruvate. Okay. So as you can see, this is the pyruvate, and it is the starting point of the synthesis of the acetyl CoA. By the help of this pyruvate, first eliminates the carbon dioxide, and the coenzyme involved in this process is the thiamine pyrophosphate or TPP. Okay, as you can see here, TPP, this coenzyme, it helps in the decarboxylation of pyruvate, and it gives us the hydroxyl ethyl TPP, HETPP. This hydroxyethyl TPP is the starting point, and the enzyme which is involved in this decarboxylation process is the pyruvate dehydrogenase. Okay, this enzyme catalyzes this decarboxylation, and TPP is the coenzyme. You need to remember there is a difference between enzyme and coenzyme. Okay, and in our previous lectures we have discussed the difference between the coenzymes and enzymes. Basically, coenzymes are needed by the enzymes to fulfill the enzymatic reactions. Okay, So this role of this coenzyme thymine pyrophosphate is the decarboxylation of the pyruvate, and then again this formation of hydroxyethyl TPP, it is followed by the next step. Okay, so in the next step the lipoic acid comes. Okay, and what is the role of this lipoic acid? Basically, this lipoic acid binds to the acetyl group of acetyl group. From where this acetyl group forms? This acetyl group forms from this part, this alcoholic part, which is derived from the pyruvate, and it is binded with this lipoic acid residue. And this from this step, the transesterification process happens. Means that the lipoic acid, it has this acetate part, and we have this. Actually, this is also a ester, thioester. As you can see, there is a C double bond OS. There is a thioester bond here. Means the lipoic acid, it is forming a thioester with this acetate moiety. And when the coenzyme A comes, the transesterification happens. What is the meaning of transesterification? It means that this acidic part, means this acetate part, it is transferred from this lipoic acid residue to this coenzyme A. Means this ester is converted into this ester, and this process is called the transesterification process. Okay. So now this transesterification process is catalyzed by this coenzyme lipoic acid, and the enzyme which is involved in this process is the dehydrolipolyl transacylase. Okay, I will suggest you not only remember the names and the functions of the coenzymes, also remember the names of these enzymes involved in this process. Like in the first process, the coenzyme is TPP, but the enzyme is pyruvate dehydrogenase. In the second step, the coenzyme involved is the lipoic acid, but the enzyme is the dehydrolipolyl transacylase. And in the third step, which is the what is the role of our third molecule, that is the FAD. FAD is the another coenzyme, and the coenzyme FAD, it is the actually it is it reoxidizes the lipoic acid. Okay, and it is just step 4. And the NAD plus, what is the role of this NAD plus? NAD plus actually reoxidizes this FADH2 to FAD. So what happens here? FAD, it reoxidizes lipoic acid and it converted to FADH2. Okay, if it is reduced to FADH2, and in the FADH2, and at the end of the catalytic cycle, this NAD plus, it reoxidizes this FADH2 to FAD and forms NADH plus H plus. So this NAD plus, it can be called the coenzyme in this process. Okay, so please remember the 4 steps of the acetyl-CoA biosynthesis. It is a very important topic, and the role of the coenzymes involved in each and every step, and also the names of the enzymes and their functions. So I hope it is clear, the biosynthesis of acetyl-CoA and what is the role of each and every coenzyme. And I will move toward now problem number 4. Okay. So the problem number 4, it says that the hydrogen which are most likely to get exchanged with deuterium when the following molecule is dissolved in D2O. So this is the structure of the molecule. So what will happen when this molecule is dissolved in D2O? There are 4 hydrogen marked here, as I will just mark it here. There is a hydrogen A, this hydrogen B, this hydrogen C, this one, and hydrogen D, this one. Okay, so when this molecule is dissolved in D2O, among these 4 hydrogens, which ones will we get exchanged with deuterium? Let's discuss, let's see the answer first, and then we will just discuss this problem. So the options of this question are number A, HA and HC, number B, HB and HD, number C, HB and HC, and number D is the HA and HD. So let us first note, okay, the correct answer for this question is HB and HC. Okay, so which one are the HB and HC? This one is the HB and this one is the HC. And let me tell you, like this HA, it will also get exchanged when this molecule is dissolved in D2O. And why these molecules get exchanged and how we can know? Actually, as you can see, this is the alcoholic proton. Okay, so basically, these alcoholic protons, these are very easy to get exchanged by the deuterium. Okay, this HB and HA, these are connected to a very electron withdrawing moiety, this thiazole group, and due to this positive charge on the nitrogen, it is very electron withdrawing, and that is why easily this Ha and Hb will also get also be replaced by deuterium when this molecule is dissolved in D2O. Similarly, oxygen is very electronegative, and that is why easily this Hc is also easily get exchanged with deuterium. But Hd, as you can see, the Hd here, it is connected to a benzene ring, and the benzene ring does not have any strong electron withdrawing force itself. So this Hd, it will not get exchanged with deuterium when this molecule is dissolved in D2O. So among these four options, the correct option will be H. Actually, the HA and HC, they I can also, so there should be two correct answers for this question. There is a mistake here in the options. Option A will also be correct, and option C also be correct. Okay, and later I will just correct it before uploading this slide PPT. Sorry for the mistake. And so let us discuss how can we know that these protons are getting exchanged in D2O. So this can be, the hydrogen deuterium exchange can be identified in the NMR spectroscopy. Okay. So one way to pick testing with the peak is from alcohol OH. Actually, the in organic molecule generally OH peaks are very easily exchanged with this deuterium. So when what happens if you add a deuteriate solvent D2 or deuteriated methanol, this hydrogen, alcoholic hydrogen, will be replaced by this deuterium, and we will not get this peak in this NMR spectroscopy for this OH. Okay. So generally the OH broad peak comes near this after above the near the 5 or 4.5 in the NMR spectroscopy, but when it is dissolved in D2O, there will be no peak of OH. Okay, because this peak, because deuterium is NMR inactive at the proton sequence. So similar thing will happen with this proton and this proton when they will dissolve in the D2O. Again, I am saying there is a printing mistake in this question. I will just resolve it before uploading this PPT, and I hope it is clear that which protons will be getting exchanged with the deuterium. So basically, the protons which are attached with the very electron withdrawing groups, they will be basically exchanged with the deuterium. Okay, when the that is added in the deuterated solvent. Okay. So next, I will move toward the next problem. So the next problem number 5, it says that which of the following medicinal class of compound is not a DHFR inhibitor? And the options are option A, trimethoprim, option B, amoxicillin, option C, pyrimethamine, and option D, methotrexate. Okay. Also, this is a medicinal class of this compounds. They all are medicinal class. Also, you know, amoxicillin is the antibiotic, and okay, there is a question. B. Okay. Yes. You are correct. The correct option of this will be amoxicillin. Okay. So, what is? Let us discuss what is the DHFR inhibitor. Actually, the DHFR is the enzyme which is called the dehydrofolate reductase. Okay, this dehydrofolate reductase, what this enzyme do? Basically, it catalyzes the reaction between the dehydrofolate and tetrahydrofolate. This dehydrofolate reductase, it actually reduces this dehydrofolate to tetrahydrofolate, and in this process, the NADPH, it is oxidized to NADP plus. That is the hydrogen from the hydride from this NADPH. It is transferred to the it is transferred to the dehydrofolate, and it is converted to tetrahydrofolate. Okay. So this is the chemical reaction which is catalyzed by this enzyme of dehydrofolate reductase. Now, what is a dehydrofolate reductase inhibitors? Actually, these inhibitors inhibit this function of this dehydrofolate reductase enzyme. So, what will happen if this dehydrofolate enzyme inhibitors are added? They will inhibit the formation of this function of this enzyme, and in that case, what will happen? The tetrahydrofolate synthesis will be inhibited, and by this what will happen, like if it is like that trimethoprim, what happens that it blocks the bacterial enzyme. Okay, means in the bacteria, it this DHFR inhibitor trimethoprim, it blocks the synthesis of this tetrahydrofolate. Okay, the pyrimethamine, this pyrimethamine is a protozoan enzyme. Okay, and this inhibits the protozoan DHFR enzyme, and in the mammals, this methotrexate is the inhibitor for this DHFR. And these all are the medicinal class of compounds. But the amoxicillin, it is not it is an antibiotic actually, but it is not a DHFR inhibitor. Okay, so remember what is the role of DHFR in the folic acid biosynthesis. This is a tetrahydrofolate. It is actually the active form of the folic acid, which is a very important vitamin in our body. And by blocking this biosynthesis, we can achieve this kind of medicinal class of compounds. And amoxicillin, it is not a DHFR inhibitor. But remember the names of these three DHFR inhibitors: this trimethoprim, pyrimethamine, and methotrexate. Okay, these are very important. So I hope it is clear from this question wise, and next I will move toward the next problem. Okay. So the problem number 6, it says that which of the following statements is true regarding the folic acid biosynthesis. Option A is it consists of pteridine ring, PABA, and aspartic acid. Option B, it says that the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthesis enzyme. Option C is the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by the dihydrofolate reductase enzyme. And option D, humans can biosynthesize folic acid. So among these four options, you need to identify the true statement regarding the folic acid biosynthesis. So let us first see the answer, and then let us discuss the folic acid biosynthesis. Okay. So the correct statement or the true statement among these four are option B, the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthesis. Okay. So let us discuss the structure of folic acid first. Okay. So this folic acid, this is the structure. From this figure you can see the structure of folic acid here. So from this structure you can identify the why an option A is incorrect, because the folic acid, the structure is divided into three parts. The first part, it is the 2-amino 4-hydroxypteridine, or it is a pteridine ring. This nitrogenous ring, it is a pteridine ring. Okay. So what is a pteridine ring? The pteridine ring, it is basically the two nitrogenous heterocyclic ring which are fused together. It is a pyrimidine ring and it is a pyrazine ring. And when a pyrimidine and pyrazine ring are fused together, they are called the pteridine ring. Okay. I told you in the last class, please remember few heterocyclic compounds, few heterocyclic cycles and their names. In the previous week 7 slides, I have mentioned few heterocyclics, and these are very important because these heterocyclics are involved in many medicinal compounds and biological compounds. So this is the structure of pteridine ring. The second part is a PABA, or para-aminobenzoic acid. The full form of PABA is para-aminobenzoic acid, which is a very easy structure. There is a benzene ring and there is a acid and amine group para to 1/4 position to each other. And there is a glutamic acid. Okay, so in the first option it is incorrect because there is a in option there is a aspartic acid, but the correct option will be glutamic acid. Glutamic acid is present in the. Okay, somebody wanted to join, I think. Okay, so the glutamic acid, it is present in the folic acid. Okay, so now, so this is the this is a very comparatively very concise and easy flowchart for the biosynthesis of folic acid, and I will suggest you please remember each and every step and the enzymes involved in this stage. So what happens first? At the first step, the para-aminobenzoic acid, this PABA, and the pteridine, they are converted, they are reacted together and converted to dihydroteroic acid, dihydroteric acid. Okay, this dihydroteric acid, it is this conversion is catalyzed by the dihydroteroate synthesis enzyme. Okay, now the next, it is the first step. Okay, this is the first step for the folic acid biosynthesis. Now what happens in the second step? In the second step, this dihydroteroic acid, it is converted to dihydrofolic acid, and the enzyme involved in this process is the dihydrofolate synthesis. Okay, so now we know why is the option B is correct, because it says that the conversion of dihydroteroic acid to dihydrofolic acid is catalyzed by dihydrofolate synthesis enzyme. Okay. Now, the option C, it says that the conversion of dihydrofolic acid and the to tetrahydrofolic acid, it is basically catalyzed by this dihydrofolate reductase enzyme. Okay, so what is and ultimately this tetrahydrofolic acid, it is the active form of the folic acid. Okay, so remember there are 3 steps for the biosynthesis of folic acid, and in each and every step there are 3 enzymes are involved. The dihydropteroate synthesis is catalyzes the synthesis, it basically synthesizes the dihydroteroic acid, and the second step is the synthesis of dihydrofolic acid, which is catalyzed by the dihydrofolate synthesis, and the third step is the synthesis of tetrahydrofolic acid, and this is catalyzed by the dihydrofolate reductase enzyme. Okay, so remember the role of these 3 enzymes and the name and the role of these 3 enzymes. Okay, for the folic acid biosynthesis. Also, please remember the structure of the folic acid. Okay. So I hope I am clear, and now I will quickly move toward the next problem. If you have any doubt, any problem regarding any question, please write in the chat box. Okay, at the end of the session, I will try to answer your query. So problem number 7, it says that which of the following enzymes convert DUMP to DTMP? Okay, and the options are A, SHT, option B is DHFR, option C is DHFS, and option D is TS. So actually, the names of the enzymes, sometimes the questions can come as like only abbreviations are given. Okay, the full name is not given. So I will suggest with the name, please remember the abbreviation of each and every enzyme. Okay, like DHFRI mentioned in the previous question that it is the dihydrofolate reductase, like DHFS, it is the dihydrofolate synthesis. Okay, so please remember the abbreviation and names of the enzymes. So which of the following enzyme converts DUMP to DTMP? Okay, I will just first show you the answer. The correct answer will be number D, TS. Now, what is the TS? It is the it is the thymidylate synthesis. Okay, this thymidylate synthesis, it converts this DUMP to DTMP. So basically, this DUMP, it is the dihydrouridine monophosphate, and the DTMP, it is the dihydrothymidine monophosphate. So this is uracil and thymidine. We all know that uracil, it is present in the RNA. Okay, this uracil, U stand for this uracil, and T stand for this thymine. And the uracil, it is a nitrogenous base which is present in the RNA but absent in the DNA. Similarly, thymidine, it is a nitrogenous base which is present in the DNA but absent in the RNA. Okay, so what happens biologically, this conversion of this between these two nitrogenous enzymes, which is it is catalyzed by the thymidylate synthesis. Okay, so this is the actually catalytic cycle. Okay, so this is connected with the folic acid biosynthesis or the folic acid cycle. As you can see, there is a this is called the salvage pathway. Okay, so what happens that the first one, the SHT, it is stand for serine hydroxymethyl transferase. What's this serine hydroxymethyl transferase does? It actually converts this CH2H4 folate to H4 folate, means tetrahydrofolate, methylene tetrahydrofolate. It is converted to tetrahydrofolate by this enzyme, and in this process, a serine molecule, it is converted to glycine. Okay, so what happens? The serine is converted to glycine, and this catalytic cycle. And next, what happens? Like this dihydrofolate is converted to tetrahydrofolate by the dihydrofolate reductase. And in this case, so what happens? Like NADPH, it is converted to NADP, and the dihydrofolate is converted to tetrahydrofolate, and this tetrahydrofolate is converted to this methylene tetrahydrofolate, and by this enzyme serine hydroxymethyl transferase. And what this enzyme do, basically it transfers one hydroxymethyl group from serine to this tetrahydrofolate, and the serine is converted to the glycine. Okay, and what this thymidylate synthesis does, it converts actually DUMP to DTMP, and this DTMP, by the help of this thymidine kinase, this is converted to DTDP and DTTP, which is ultimately gives us the DNA biosynthesis. Okay, So you don't need to remember each and every step for this DNA biosynthesis and all this. Just remember what is the role of each and every enzyme. So, okay, I'm saying that it is very important to just know, identify what is the role of the enzymes, because this is the, you don't need to remember each and every step, but remember what the enzymes are doing. Like the SHT, it is converting tetrahydrofolate to this methylene tetrahydrofolate, and in this step, one serine is converted to glycine. Like the dihydrofolate is converted to tetrahydrofolate by the dihydrofolate reductase, and this thymidylate synthesis, it is converting DUMP to DTMP. Okay, just so remember the roles of each and every enzyme, and it will be enough. Okay. So quickly, I will move toward now problem number 8. It says that which of the following correctly pairs with the coenzyme with the group transferred by the coenzyme. Okay, the option A is the retinal and phosphoryl group. Option B is biotin and carbon dioxide. Option C is ATP and methyl. And option D is thiamine pyrophosphate and transamination. Okay, so I will show you the answer and then we will discuss. So the correct option is the biotin and carbon dioxide. Biotin is the coenzyme and it transfers this group of carbon dioxide. Okay. So this is the structure of the biotin. So this is the reaction catalyzed by the biotin. So what happens? Somebody wants to join, I think. Okay. So what happened? Yeah. So I was in this problem. So basically, biotin catalyzes the transfer of the carbon dioxide groups to the substrate. Now, as you can see, there is this reaction which is catalyzed by biotin, and what happens in this reaction? The pyruvate, it is converted to the oxaloacetate. Now, as you can see from the structure, this is the structure of this pyruvate, and there is a carbon dioxide attached here. This extra carbon dioxide, this group is transferred by this coenzyme biotin. Okay, and this pyruvate is converted to this oxaloacetate. This is the part from the pyruvate, and after joining of this carbon dioxide, it is converted to oxaloacetate. And from where this carbon dioxide group is coming? It is coming from this bicarbonate residue of the ATP, and in this case, one ATP is converted to the ADP, okay, and inorganic phosphate. So ATP and bicarbonate is converted into ADP and inorganic phosphate, and in this case, this carbon dioxide group is transferred from the transferred to the pyruvate molecule, and this reaction is catalyzed by biotin. In the next step, you can see the catalysis, the catalytic cycle. So what happens? There is a pyruvate molecule, and what happens at first? The decarboxylation of this biotin, it is taking up this carboxylate COO minus CO2 minus group from this ATP and PI ATP molecule, and it is then after this reaction, what happens? Like this carbon dioxide group, it is transferred to the pyruvate. There is a detailed mechanism, and I hope when you just see the mechanism and you will understand how it is happening. So first, basically, there is a resonance here, and from the elimination of this carbon dioxide, there is a negative charge on oxygen. When it is coming back to this forming the C double bond O group, the nitrogen, it is taking up this hydrogen from this pyruvate, and this negative charge, after resonating with this carbonyl group, it is attacking this carbon dioxide, and the oxaloacetate is forming. Okay, so this is the diagram. You don't need to remember, you don't need to memorize the scheme, just understand how it is happening, and just remember what is the coenzyme, name of the coenzyme, and the group transferred by the coenzyme. Okay, so I think it is clear, and I will now move toward our next problem. So problem number 9, it says that which are the following is or are vitamin K antagonist. Okay, the options are dicoumerol with warfarin. Option B is warfarin. Option C is menadione, and option D is both A and B. Okay, so let us first see the answer, and then let us discuss what are the vitamin K antagonists. Okay, so yeah, both A and B are the vitamin K antagonists. So what is the vitamin K antagonists and what they do? Actually, vitamin K, what vitamin K helps us actually in the blood clotting. Okay, all you know that when we are hurt at some point, or there is bleeding, After some time the bloods are they form a very solid state, and there is a bluish clot in our body, and that is because of the vitamin K. The vitamin K actually does this kind of work. So what happens is what this vitamin K antagonists do, they basically rupture this vitamin K cycle. Okay, so if the vitamin K cycle is ruptured, the blood clotting will be inhibited. So why we need to inhibit this blood clotting? I mean, we know that blood clotting is needed by our body because if we are hurt at some time, or there is a bleeding. But sometimes we need this vitamin K antagonists, or they also known as a blood thinners. Okay, or anticoagulants. So why we will need this anticoagulant or blood thinners? So basically what happens, like when a patient is when there is a hypertension patient, okay, or there is a with very much cholesterol in the body, what happens, this cholesterol is this is they are stuck in the blood vessels. Okay, so if the blood vessels are narrowed by this cholesterol, so what will happen, there will be extra blood pressure in the vessels, and that is why mainly the heart blockage or heart attack or cerebral attacks, these kind of problems happen. Okay, so for this we need the vitamin K antagonists or blood thinners. In some patients the doctors give this vitamin K antagonist as blood thinners. So these two molecules are the examples of vitamin K antagonists. So what they does, that this is a vitamin K cycle. Okay, so as you can see that there is a vitamin K hydroquinone, which is transferred to vitamin K epoxide, and there is a enzyme, there is a gamma glutamyl carboxylase enzyme, and after this, the vitamin K epoxide is converted to vitamin K, which is catalyzed by vitamin K epoxide reductase enzyme, and at the in this third step of catalytic cycle, the vitamin K is converted to vitamin K hydroquinone by this DT diaphorase. Okay, again, if you if possible, remember the names of the enzymes involved in the vitamin K cycle, and also the names of the vitamin K antagonists. Okay, so the names of the vitamin K enzymes, these are the gamma glutamyl carboxylase, vitamin K epoxide reductase, and DT diaphorase. And vitamin K antagonists, they will do, they will basically inhibit this enzymes. Okay, like in previous some in one slide we saw that the DHFR inhibitor, they inhibits the dehydrofolate reductase enzyme, and vitamin K antagonists, these are also inhibitors of these enzymes involved in the vitamin K cycle. And the examples are these two molecules, and these are all vitamin K antagonists. So remember the names of this vitamin K antagonists and what they do, and this vitamin K cycle. Okay. So the problem number 10, it says that the enzyme which catalyzes fibrinogen to fibrin conversion is number A, trypsin, number B, thrombin, number C, prothrombin, and number D, pepsin. Okay. So the fibrinogen to fibrin conversion, it is also a part of the vitamin K and the blood clotting mechanism in our body. So let us just let us just see the answer first, and then we will discuss. So the correct answer, the enzyme which catalyzes fibrinogen to fibrin conversion is thrombin. So yeah, this is the basically the mechanism, the schematic diagram of this blood clotting or the clot formation, and in this body. So what happens, like this is the mainly the in our what happens in our body, this flow chart. So what actually happens, like there is a prothrombin, okay, and prothrombin activator with the help of this calcium 2 plus, which forms the thrombin. This thrombin actually catalyzes the fibrinogen to fibrin conversion, and this fibrinogen actually converts to the fibrinogen monomer to this by this help of this thrombin, and thrombin again is it after with help of this calcium 2 plus, it forms fibrin fibers, and this fibrin fiber are basically forms some cross linking polymers, as you know that they're like there is a fibrin, these are the monomer, and when they form the cross linking polymer, after this polymer, this forms some gel type of thing, which actually shows you in the like a clot. Okay, so this thrombin is the activated fibrin stabilizing factor, which catalyzes the fibrinogen to fibrin conversion. So what happens, like in our body, the platelet aggregation happens, and if there is a any injury, And the activation of coagulation, the first step is the clot initiation, and the clot formation. This is this thrombin. It catalyzes the fibrinogen to fibrin conversion, and the fibrin monomers converts this to fibrin polymer, and the fibrin fragment. It is fibrinolysis. Fibrinolysis means when the clot will heal. This when this injury will heal, this clot will be diminished. Okay. So this is basically, this all reaction is, it's catalyzed by the vitamin K, and I think remember what does the thrombin do, and what is the role of this in the vitamin K function. Okay. So I will move toward the problem number 11. If you have any doubt regarding any question, please write in the chat box. At the end of the session, I will try to answer those queries. And problem number 11, it says that the cobalamin is a coenzyme which is derived from the vitamin B. Option A is vitamin B5. Option B is vitamin B6. Option C is vitamin B12. And option D is vitamin B2. And so the correct answer of this problem is option C, vitamin B12. So we all know that cobalamin is a coenzyme of vitamin B12, and it is the structure of the vitamin B12. Okay. So this is the structure of vitamin B12, and there is a, we'll just quickly discuss the structure. So hence this is a nitrogenous ring. You can see there is a this is the, there are 4 nitrogen. Okay, there is one nitrogen here, another here, another here, another here, and there is the macrocyclic ring. It is called the coring ring. Okay, this question also can come. This, what is the ring present in the vitamin B12? So remember, it is a coring ring, and there is a 5-member ring which are connected by a carbon. There are 3 1 carbon bridge, and one in one of these 5 rings are there joint. There is no carbon in between. Okay, so it is a 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 carbon member ring. Okay. So there are there is this is macrocyclic ring is called corin ring, and there is a cobalt plus one. This is very important. There is a cobalt plus one present in the center of this corin ring, and it is chelated with this 4 nitrogen atom of this corin ring at this in this equatorial plane, and the in this two axial plane there are 2 groups present at the there up to the front there is a cyanide group there, that is why it is called cyanocobalamin, the vitamin B12, and there is a cyanide group present here, and in this stage in the axial group there is a heterocyclic molecule, and there is a sugar moiety also is present. So you don't need to remember the each structure, don't need to memorize at all the structure of this huge vitamin B12, but remember there is a corin ring present in the vitamin B12. Okay, in the center there is a cobalt plus one. It is very important to remember the oxidation state of the metal ion. So there is a cobalt plus one present in vitamin B12, and the name of the ring is the corin ring. And this corin ring, there in the equatorial plane there are 4 nitrogen, and at the axial plane there is a cyanide group is present. Okay, so just remembering this 3 point is going to be enough for this vitamin B12 structure. You don't need to memorize the whole structure of this vitamin B12. Okay, so now I think it is clear the structure of vitamin B12 and the groups are present. Now then I will move toward the problem number 12. Okay, so problem number 12, it says that which of the following statements regarding NAD plus is incorrect. Option A, it contains a nicotinamide ring which accepts a hydride ion during reduction. Option B, it reoxidizes FADH2 to FAD in the last step of acetyl-CoA synthesis. Option C, it contains ATP as a part of, there is a, okay, we were in the option. Okay, again I will repeat the problem number 12. It says that which of the following statements regarding NAD plus is incorrect. Option A, it contains a nicotinamide ring which accepts a hydride ion during reduction. Option B, it reoxidizes FADH2 to FAD in the last step of acetyl-CoA synthesis? Option C, it contains ATP as a part of its structure, and option D, it contains a diphosphate linkage. So I will just first show you the answer, and then we will discuss what is the NADH plus and what is its role, and why which the options are correct or incorrect. Okay. So the answer of this problem number 4 is the option C. It contains ATP as a part of the structure. It is the incorrect statement among the followings regarding NAD plus. So this is the structure of NAD plus. Okay, this is the structure of NAD plus, and as you can see, okay, there is a better structure for this NAD plus. So it is okay. Just one. So there is a nicotinamide group present in this NAD, and this is the nicotinamide group. This is the nicotinamide group. Okay, and when the reduction happens, it is the oxidized form NAD plus. Okay, basically it acts as a redox coenzyme. You can see, and it is the redox part, the reduced part or redox part. It is the NADH, and the NAD plus is the oxidized part. And you can see there is a this is the nicotinamide ring. This ring is called the nicotinamide ring. Okay, so in the reduced part there is a two hydride ions are two hydride hydrogens are present here, and when the reduction happens, that is it is when the NADH it oxidizes, it reduces some molecule and get oxidized to NAD plus. The hydride from this nicotinamide ring, it is transferred. Okay, this hydride from this nicotinamide group, it is transferred to some substrate. That substrate gets reduced and it gets oxidized to the NAD plus. Okay, it reoxidizes NADH2 to FADH2 to FAD at the last step of acetyl-CoA synthesis. Now, if you remember in problem number 3, I think, we discussed the acetyl-CoA biosynthesis. And in the last step, I told you that the conversion of the FADH2 to FAD, it actually happens with the help of the NAD plus. Why? The FADH2 to FAD plus, it happens, okay, at once. Let us just okay. So what happens, like I think if my voice is clear or the presentation is visible, can somebody just write in the chat box? I didn't check today, but if you can hear my voice or see the presentation clearly, please write in the chat box. Okay. So what happens, like in NAD plus, in the last step of this acetyl CoA biosynthesis, somebody has written something. Okay, thank you so much. That it is. I'm glad it is clear. Now we will discuss. Like if you remember in the previous some slide, I will just show you the slide. I think it is slide number 3 where we discuss. Yes. So as you can see here, we discussed the acetyl CoA biosynthesis, and I said that at the third and fourth step actually, the FAD, it actually deoxidizes the lipoic acid and it is converted to FADH2, and this FADH2 it is reoxidized by this NAD plus to FADH. Okay. So how it is happened, and now you know that the NAD plus, it will just accept the hydride from this FADH2 and it will convert it to NADH, and it will convert it to NADH, and the FADH2 it will get quickly move, yeah, here. So at the last step, it actually what happens, like this NAD plus, it accepts the hydride from this FADH2 and it is converted to NADH, and FADH2 is reoxidized to FAD. Okay. Now the option C says that it contains ATP as a part of a structure, which is false, because this, as you can see, the NAD plus structure, there is no ATP present. Okay, here no ATP is present, but here is a diphosphate linkage is present here. Okay, as you can see clearly from the structure that there is a diphosphate linkage which is here. This diphosphate linkage is present here. So the option C, it is incorrect. There is no ATP in the structure, but there is a diphosphate linkage. So remember that NAD plus, it contains a nicotinamide group which participates in the hydride donation during reduction, and there is a diphosphate linkage present in the structure. And just if possible, please remember the structure of NAD plus. Okay, it is very important. And because NAD plus, it is a very important for there are many bioprocesses where it helps in the oxidizing and reducing the substrates. Okay. Now, quickly we will move toward the problem number 13. It says that which of the following statements is false regarding ascorbic acid? Option A, it is a strong antioxidant. Option B, it catalyzes hydroxylation reaction. Option C, it is produced inside human body, not required through diet. And option D, it chelates with iron to make it more soluble. So what will be the false statement regarding the ascorbic acid? The ascorbic acid, we all know it is actually the vitamin C. Okay, vitamin C, it is known as the ascorbic acid. Vitamin C. So what will be the incorrect statement among these four regarding the ascorbic acid? It will be the number C. It is produced inside our body, not required through diet. Vitamin C or ascorbic acid, it is not produced in our body. Okay, it is we require to take vitamin C from the food substance, and all we know that the citrus fruits like lemons and oranges, they actually give us this vitamin C. Okay. Now, this is the structure of this ascorbic acid or vitamin C. Now, we all know that what is the vitamin C foods which we get the vitamin C. These are the citrus fruits like lemons and oranges. And what is the symptoms of the deficiency of vitamin C? This is basically the fatigue and depression, increased susceptibility to infection. Vitamin C is very necessary for us for our immunity system. So that's why in the season, during the season change, we are told by doctors to have the seasonal fruits, mainly the citrus fruits. Muscle weakness, joint and body aches, rough and dry skin, scurvy and bleeding of gum, and or loose teeth. Okay, these are the vitamin C deficiency signs. So if vitamin C is produced in our body, then we will not get this vitamin C deficiency. Okay, so the statement C is incorrect regarding the vitamin C. So we need to take ascorbic acid from through our diet. So it is a strong antioxidant. Remember, these statements are regarding the vitamin C. It is a very strong antioxidant, and it catalyzes the hydroxylation reaction. And number C is incorrect, and it chelates with iron to make it more soluble. Okay. So actually, vitamin C and iron, they are very correlated. Like vitamin D and calcium, they are correlated to each other, and we need vitamin C. And for iron intake in our body, we actually need the vitamin C. Okay, because for absorption of iron to our body through diet, we also need to take vitamin C. It is very important. Like for calcium absorption in our body, calcium ion absorption, we need the vitamin D. So you have seen actually when there is a bone related very deficiency, it is said to intake vitamin D also. So because vitamin D, it increases the absorption of calcium in our body, and vitamin C, it increases the absorption of iron in our body. Okay, so I think it is clear, and quickly I will move toward the problem number 14. It says that identify the incorrect statement among the following. Option A, reduction of the flavin ring disrupts the conjugation. Option B, reduced coenzyme are more colored than the oxidized one. Option C, in FAD system, all the oxidation reduction chemistry takes place on the flavin ring. And option D, FADH2 is the reducing agent. So what will be the incorrect statement among the following? The incorrect statement will be option D. Option B, reduced coenzymes are more colored than the oxidized one. So let us first see the structures and okay. So if this question is basically related to the FAD or flavin, to the FAD, which we discussed in our previous questions, like it is important coenzyme in the acetyl CoA biosynthesis and many other biological reactions in our body. So basically, in FAD system, there is oxidation reduction chemistry happens. Just like NAD plus, this FAD also, it is involved in the oxidation reduction chemistry, and the oxidized form, it is known as the FAD. Okay, and the reduced form, it is known as the FADH2. And this FAD, upon the addition of the two hydrogen and two electrons, it is converted to the FADH2. Okay, and what happens like for this reaction also, as we know, this reaction is can be catalyzed by NADH, and NADH is converted to NAD plus. Okay, just FAD can converted to FADH2 can be converted to FAD by NAD plus. and also FAD can be converted to FADH2 by NADH. So basically these are very interrelated to each other. Okay, there is basically simple oxidation and reduction chemistry, and only the transfer of hydrogen from one part to another part. Okay. So, the first question says that the reduction of flavin ring disrupt the conjugation. So, it is the correct, because as you can see, there is a conjugation here. As you can see, there is nitrogen, double bond, single bond, and then again double bond nitrogen. So, this is basically the conjugation. Okay. And this conjugation upon reduction, when there is a NH and NH, so there is no conjugation. This conjugation means there is a double bond, single bond, double bond. This is called the conjugation. But upon the reduction, there is no conjugation. There is no conjugation present. So the reduction in of the flavin ring disrupts the conjugation. It is the correct statement. Now, second question, it is says the reduced coenzymes are more colored than the oxidized one. It is actually the incorrect, because more the conjugation, more will be the color. In the reduced enzyme, there is no conjugation, so there will not be more colored, or they will be less colored than the oxidized one. The oxidized one, due to more conjugation, will be the more colored. The third option, it is correct, because the oxidation reduction, it is taking place in the flavin ring, and the FADH2 is the reducing agent. We all know it is correct, because FADH2 is the reducing agent, and it reduces substrates and get oxidized to FAD. Okay. So I think it is clear. This problem number, quickly I will move to our last problem of today's class. So the problem number 15, it says that which of the following statements regarding mature prothrombin is true? Option A, mature prothrombin has 10 of the glutamyl residues in first to 42 residues from amine terminus. Option B is the carboxylation process is vitamin K independent. Option C, mature prothrombin, the glutamyl residues are modified as gamma carboxyglutamyl residues. And option D, gamma carboxyl residues are good chelators for the magnesium ions. So what will be the correct statement regarding this prothrombin, mature prothrombin? It is the option C. The mature prothrombin, the glutamyl residues are modified as gamma carboxyglutamyl residues. So let us so this is the reaction. Okay. So why the first option? The first option says that the mature prothrombin has 10 of the glutamyl residues in first 42 residues of amine terminus. It is false, because it will be immature. Okay, the immature prothrombin actually has the 10 of the glutamyl residues. Okay. And the carboxylation process, it is vitamin K independent. It is also incorrect, because it is vitamin K dependent. Okay. Option C is correct, and we will discuss it later. And option D, it says that gamma carboxyglutamyl residues are good chelators for the magnesium ion, and it is also incorrect. It will be calcium ions. Okay. So if you read the vitamin K part carefully about the mature prothrombin, you can easily identify which will be the correct statement among these four. So this is the actually the conversion. The glutamate residue close to NH2 terminal group of prethrombin. This is the glutamyl residue, which is the close to NH2 terminal group of the prethrombin, and this is the gamma carboxyglutamyl residue. Okay, in mature prothrombin, this glutamyl residue, it is modified as gamma carboxyglutamyl residue, and the enzyme which catalyzes this reaction is the gamma glutamyl carboxylase enzyme. Okay. So this is also related to the vitamin K enzyme of the vitamin K and the blood clotting mechanism by this vitamin. Or we in the previous slides just discussed that prothrombin is converted to thrombin, which is in turn is converted to the converts fibrinogen to fibrin. And so remember, like in the why this options are incorrect. The option A, the actually it will be the mature, it will be the immature prethrombin, which has the 10 of the glutamyl residues, and carboxylation process is vitamin K dependent. In mature prothrombin, glutamyl residues are modified as gamma carboxyglutamyl residue, and option D, it is the good chelator for the calcium ion, not for the magnesium ion. Okay. So that's it. These were the 15 questions related to your week 8 course content. And today it was the if you have any doubts today regarding today's class, I will just share my email ID with you. So if you have any doubt regarding to today's class, please do write an email to me. Okay, in the chat box, just I am typing. So this is my email ID. If you have any doubt regarding today's class, at any question, any point, please do write an email to me. This lecture video and this PowerPoint presentation will be shared to you by the NPTEL site. In the NPTEL website, you can find this problem solving session. And each week we conduct this kind of problem solving session on Friday from 7 to 8 p.m. And see you next week regarding the week. It will be the week nines, and we will discuss the week nine session. And I hope today you learned something about this and understand this course better. And so thank you for joining today's class, and I will see you in the next class. Okay. Now I will close this session. And if you have any doubt regarding to any problem and today's class, please write an email to. I will see you in the next lecture, which will be on next Friday from 7 to 8 p.m. Okay. Thank you so much. Thank you so much. Now I will close the meeting.